We're going to be starting on Chapter 3, which is the cellular level of organization. Now we will be covering quite a bit in this chapter, but these things include the parts of the cell. We're going to be looking at the plasma membrane and the cytoplasm. We will be going over mitosis and meiosis, and then a little bit of the effects of aging on the cell. So cells are actually the smallest units of life. Cells come in many different types and forms, like neurons, muscle cells, blood cells, just to name a few different types of cells. Now, regardless of the differences between cells, they all have the same basic parts and the same common functions of those parts. For this reason, it really is possible to speak of a generalized cell. So a generalized cell can be split or subdivided into three different parts. These are the plasma membrane, the cytoplasm, and the nucleus. So the cytoplasm includes the cytosol, which is kind of like the gel surrounding all of the organelles, and the organelles are the second part of the cytoplasm. And the nucleus includes chromosomes, and those chromosomes have genes. Now we will be going over all of these parts of the cell one at a time. So this is kind of like a really nice overall view of the entire cell with all of its components. I won't go over all of these in this picture, but feel free to come back to this slide and reference once we've talked about a different organelle or talked a little bit about the nucleus, that kind of thing. You can kind of see it in its pictorial form here. So first we're going to discuss the plasma membrane. Now the plasma membrane does have several functions. The first of which is that it acts as a mechanical barrier. It's just another way of saying it acts as a physical barrier to keep things in and to keep other things from entering. Second function is that it has selective permeability. So this means that it allows some things in and not others. It also creates an electrochemical gradient. And we will go over this in a little bit more detail as well a little bit later. Electrochemical gradient just means that it can maintain differences in concentrations of ions and molecules on either side of the membrane. So it can maintain a higher concentration of sodium ions, for example, maintain a higher concentration outside the cell than inside the cell. So it has kind of a gradient across the membrane. The fourth function of the plasma membrane is communication. Communicating mainly with other cells, with proteins that are in the plasma membrane. And then our fifth major function of the plasma membrane is cell signaling. Okay, so let's look at the plasma membrane itself, this little picture here on the right. So the plasma membrane is made up of about 75% of a phospholipid bilayer. So this occurs, remember, because those lipids are amphipathic. So remember, they are amphipathic. So remember, this means that they have both polar and non-polar parts. So they have, it's not really labeled here, but they have these polar heads. These are actually going to be on both sides here. And then here on the inside are going to be these lipid, we'll just put, these are non-polar tails. About 20% of the membrane is going to be these cholesterol molecules here. Now, just like phospholipids, cholesterol is also amphipathic. Remember, we kind of discussed it's kind of weakly amphipathic, but we do still consider it amphipathic. Now, cholesterol is going to essentially wedge its plate-like hydrocarbon rings between the phospholipid tails, and cholesterol works to stabilize the membrane. So this is going to be one of the biggest functions that cholesterol has in the plasma membrane is stabilization. Stabilizes the membrane. So normally this membrane is going to be very fluid, so it kind of acts as ocean wave, you can kind of think of it like, but cholesterol is going to stabilize that membrane. So what that means is that it decreases the mobility of the phospholipids. It decreases the fluidity, you could also say. So more cholesterol, or like the ratio of cholesterol to phospholipids, if there's more cholesterol in the plasma membrane, it's going to be less fluid. Whereas if there's less cholesterol, then it's going to be a much more fluid plasma membrane. Then about 5% of the plasma membrane are going to be glycolipids. So these are labeled here. These are lipids with attached sugar groups. Really, they're only found on the outer plasma membrane surface. So like I said, the plasma membrane is a very fluid membrane, and it's really best explained by using a structural model called the fluid mosaic model. Now, according to this model, the molecular arrangement of the plasma membrane resembles a continually moving sea of fluid lipids that contain a mosaic of many different proteins. So here are kind of the couple different proteins that we see in the membrane. So first is integral membrane proteins. Now these extend into or through the lipid bilayer. So kind of like here, this channel protein, it sees both the external surface here and the internal surface. So it kind of crosses through the entire membrane there. Most of these are specifically called transmembrane proteins, and this means that they span the entire membrane and they protrude on both sides, like I said. Just like membrane lipids, these integral membrane proteins are amphipathic. They have to be amphipathic because they are going to interact both with very hydrophilic regions on the outsides here and very hydrophobic regions on the inside of the membrane here. So they have to be very amphipathic as well. Then we have peripheral proteins. Now, like their name implies, they're not as firmly embedded in the membrane like the integral proteins. These peripheral proteins, we can see one right here, these are attached just to the polar heads of the lipids, or sometimes they're also attached to the integral proteins at the inner or outer surface of the membrane. Now, these peripheral proteins are almost always just going to be hydrophilic, just because they're on the outside attached to the polar heads or just attached to the integral proteins. So they don't really go into the inside of the plasma membrane. They don't enter into that lipid, very hydrophobic region of the membrane. Membrane proteins largely reflect the functions that a cell can perform. We have several different membrane proteins here. The first one here is an ion channel. Now, ion channels are integral, and they form a pore through which a very specific ion can flow through to get across the membrane. These are going to be very specific. They can't just let anything through. There are very specific ion channels for specific ions. So you would see a sodium ion channel that only allows sodium through its pore. Next, we have carrier proteins. These are also going to be integral membrane proteins. And they transport specific substances across the membrane, and they do so by undergoing a change in shape. So kind of acting like scissors or a seesaw. They're going to be open here. The ion or whatever specific substance enters, they change shape and they open to the other side of the membrane, moving that substance across the membrane. Next is a receptor, also an integral membrane protein. And receptors recognize a specific ligand. Now, a ligand is just kind of another special name for a specific ion or molecule that binds to a receptor. Now, ligands are going to alter a cell's function in some way. An example used in the textbook is the antidiuretic hormone. It binds to receptors in the kidneys and changes the water permeability of certain plasma membranes. Next, we have enzymes. These can be integral or peripheral, and they catalyze reactions inside or outside the cell. This is really depending on which side the active site faces. Then we have linker proteins. These again can be integral or peripheral. And their function is to anchor filaments inside and outside the cell, really just providing a lot of structural stability and even shape to the cell. Then we have cell identity markers. These are going to be glycoproteins that distinguish your cells from the cells of someone else. Major histocompatibility complex proteins, and these are also called MHC proteins. These are a specific type of cell identity marker. These are definitely going to come up a little bit later, more so when we talk about the immune system. But these are going to be the main type of protein that are cell identity markers that identify your cells from others' cells. Now, typically there are three factors that bind cells together. Glycoproteins, these act as an adhesive. Wavy contours of the membrane, they're really going to fit adjacent cells together in kind of a tongue and groove type fashion. And also special cell junctions. Now, of those cell junctions, we have three types. So these are going to be here. So those three types of cell junctions are going to be tight junctions, gap junctions, and adhesion junctions. First, we have tight junctions on the left here. They are really impermeable junctions that essentially form continuous seals around the cells. And this prevents molecules from passing through the intercellular space. So we have this really small intercellular space here, pretty much doesn't allow anything to go between there. Next, we have the gap junctions. These are going to be communicating junctions that allow ions and small molecules to pass between them. And they pass between these little membrane channels. These types of junctions are really particularly important for communication in heart cells and also in embryonic cells. Lastly, we have the adhesion junctions. This particular adhesion junction has a special name. It is called a desmosome. It's a type of adhesion junction. And this desmosome is an anchoring cell junction that binds adjacent cells together and acts pretty much like cellular Velcro. So it has these little extracellular filaments that Velcro these cells together and are anchored on the inside by these filaments and also by plaque. So just a couple different ways that cells are held together here. Now, membrane fluidity depends both on the number of double bonds in the fatty acid tails of the lipids that make up the bilayer, number of double bonds. So remember when we talked about the differences between saturated versus unsaturated fatty acids. And it also depends on the amount of cholesterol present. So these are going to be the two main things that membrane fluidity depends on. So remember, each of these double bonds puts a kink in the fatty acid tail, and each of those little kinks increases the membrane fluidity by preventing the lipid molecules from packing really tightly together. So if we had these lipid tails here in the plasma membrane, remember this is going to be a bilayer, so we have two layers here, but we'll just look at half of it for now. So if they are completely saturated, these tails are going to be completely straight. We can kind of pack them really tightly together. On the other hand here, if we start having kinks kind of sticking out here, kind of can't pack them as tightly together. And then especially here, if we have another kink here, and then we have a kink on this side kind of thing, maybe a kink here, it's getting to be really kind of chaotic, and they can't pack very tightly together. So when we can't pack them very tight, then they are further apart from each other, and that really increases that membrane fluidity. And then there's also the cholesterol. So remember, cholesterol works to stabilize the membrane. So increasing the amount of cholesterol in a membrane is going to decrease the fluidity. It makes it more rigid. Now, membrane fluidity is really a big compromise for the cell. This is because a very rigid membrane is going to lack a lot of mobility, and a completely fluid membrane is going to lack the mechanical support as well as structural organization that's required by the cell. So really we have to have kind of a good balance between fluidity and rigidity. Now, it was very briefly mentioned before, but the plasma membrane is also selectively permeable. Now, the term permeable means that the structure permits the passage of substances through it, while impermeable means that a structure does not permit the passage of substances through it. So permeable means that we're allowing things through. Impermeable means we're not allowing anything through. So the membrane being selectively permeable, it means that it lets some things through and not others. So the plasma membrane is going to be very highly permeable to non-polar molecules, things like oxygen, carbon dioxide, steroids, and then it's going to be moderately permeable to very small uncharged polar molecules like water and urea. And it's going to be completely impermeable to ions and to very large uncharged polar molecules. So things like glucose. Glucose cannot pass through plasma membranes at all without help. Now, because the membrane is selectively permeable, this allows the cell to maintain different concentrations of certain substances on either side of the plasma membrane. So what we call a concentration gradient, this is going to be that difference in concentrations of chemicals on either side of the plasma membrane. Many ions and molecules are going to be more concentrated in either the cytosol inside the cell or in the extracellular fluid. For example, they're going to have or in most cells, they have a higher concentration of sodium ions in the extracellular matrix around the cell and more potassium ions inside the cell. The plasma membrane also creates a difference in the distribution of positively and negatively charged ions between the two sides of the membrane. So typically, the inner surface of the plasma membrane is more negatively charged, and the outer surface is more positively charged. So when we kind of look at just a typical round cell like this, we have a nucleus here. So typically, we're going to have the inner surface of the plasma membrane being more negatively charged. There's a lot of anions that are stuck inside the cell. And then on the outside of the cell, it's going to be more positively charged. This is typical. So this kind of, again, a gradient of these differences in charge on either side of the membrane. This has its own special name as well. And this is called the electrical gradient. Together, the concentration gradient and the electrical gradient kind of make up this collective, which is called the electrochemical gradient. Now, typically, a substance will want to move down its concentration gradient or move from a place of higher concentration to a place of lower concentration. Now, this is really the natural progression of movement. Essentially, we can kind of think of it as a hill or kind of like a graph like this. And this kind of be, for example, this can be like concentration. So this is going to be the concentration of a particular ion or molecule. We'll just say, for example, this could be concentration of sodium ions. So here, where there's going to be high concentration of sodium ions, for example, on the outside of the cell, and here where we're going to get a low concentration of sodium ions, for example, on the inside of the cell, ions will always want to move down their concentration gradient. So we can kind of think of it as they want to roll down the hill. And so all ions, they will always want to move from areas of high concentration and naturally will want to move to areas of low concentration of their particular ion. So in this case, high concentration of sodium, those sodium ions will want to move to areas where there's not very much sodium. Kind of like it is too crowded in the high concentration areas. So it's kind of like if we have all these little sodium ions in an area, and then in the area right next to it, right here, there's only a couple. So this is high concentration, and this is low concentration. So this particular sodium, I'll choose a different color here. This particular sodium ion is going to naturally want to move this way. It's less crowded, and it's kind of going to be a lot more comfortable. So it's going to be from an area of high concentration to low concentration. And we just call this moving down their concentration gradient. Now, in the same way, a positively charged ion will tend to move to more negatively charged areas, and vice versa for negatively charged ions. So in this way, the concentration and electrical gradients are going to influence the movement of ions. So together, the electrochemical gradient is going to be a major factor that influences the movement of ions. Well, ions and molecules altogether. Now, transport of materials across the plasma membrane is really, really essential for the life of a cell. Now, substances generally move across cellular membranes by transport processes that can be classified as either passive processes or active processes. And this really depends on whether they require cellular energy to move. So active processes, these are going to require ATP. Whereas passive processes, they will just occur. They don't need any ATP in order to move a substance. So passive processes, really, they just move down their concentration gradient or down their electrical gradient to cross a membrane, and this is just using their own kinetic energy. They don't need an additional input of energy to move down their concentration gradient. In active processes, this cellular energy in the form of ATP is required to drive the substance uphill, or we can also think of it as driving a substance against its concentration or electrical gradient. So in active processes, we can kind of still think of this little hill that we saw. So in this case, we have high and low. We're going to be wanting to move a cell uphill, or an ion or a molecule uphill, up its concentration gradient. So let's first discuss the passive processes. So first we have simple diffusion. So diffusion, this is a passive process in which the random mixing of particles in a solution occurs because of the particle's own kinetic energy. So it's driven by the molecule's own kinetic energy. Now, if a particular solute is in really high concentration, it will diffuse toward the areas of lower concentration until those particles are evenly distributed throughout, and this creates an equilibrium. So here we can kind of see this really high concentration down here of this blue substance, so these blue particles in solution. And up here, there's going to be a very low concentration. Kind of see no blue in this area. So they will naturally want to move down their concentration gradient, going this way, kind of distributing through the entire area, making an equilibrium across the entire solution here. So simple diffusion, this is going to be a very passive process. And this is where substances will freely move through the lipid bilayer of the plasma membrane. And this is without the help of any other membrane proteins or anything like that. The most typical molecules that are going to be able to move by simple diffusion are going to be nonpolar, hydrophobic molecules. These are going to be able to cross the lipid bilayer without help. Now, there are a couple of things that can influence the rate of diffusion. So the steepness of a concentration gradient is going to be one. So say, for example, we have, we could just say like 100 molecules of a substance in one area and 0 in another area. It's going to move pretty quickly in order to kind of diffuse across the entire area here. But on the other hand, if we have, say, 10,000 molecules and then 0, also another area, it's going to move much faster. So this is a steeper gradient. Kind of a larger difference in concentration between the two values. Temperature is also going to be a factor that influences diffusion. If we have a higher temperature, it's going to increase the rate of diffusion. So it's going to occur faster if it's warmer. And then a couple of other things that would influence in a lesser way would be mass, surface area, and diffusion distance. So heavier molecules are going to diffuse slower. If there's a larger surface area to cover, it will also be slower. And if there's a larger distance in order to, or needed to diffuse across, that will also be a slower process as well. But the main processes that influence diffusion are going to be the steepness of the gradient and temperature. Next, we have facilitated diffusion. So this is a type of diffusion that's needed for molecules that are too polar or they are too highly charged to move through the lipid bilayer by simple diffusion. So this is where we have an integral membrane protein that assists the substance across the membrane down its concentration gradient. So a couple different types of facilitated diffusion that we will go over. First one here is channel-mediated facilitated diffusion. So this is where a solute will move down its concentration gradient across the membrane, but it needs a channel to go through. So it can't just go straight through and diffuse through the plasma membrane. It requires a channel protein. So it's just a helper protein to help it cross the plasma membrane. Now, most membrane channels are ion channels, and these are going to be integral membrane proteins that allow the passage of really small inorganic ions that are too hydrophilic to penetrate the nonpolar interior of the lipid bilayer. So essentially, they just allow for this small area, this kind of a pore to move through to go down their concentration gradient. So they're still moving from an area of high concentration to an area of low concentration of potassium in this case. So it's down its concentration gradient, so it is a natural movement, doesn't require any energy input. Just needs a little pore to move through. Next, we have carrier-mediated diffusion, and this occurs when a transporter is used to move a solute down its concentration gradient still, and still no energy is required in this. So in this type of diffusion, the solute here, which is going to be glucose in this example, it binds to a very specific carrier on the side of the membrane, and then it's going to be released on the other side after the carrier protein undergoes a change in shape. So this is going to be the really big important part of this carrier-mediated facilitated diffusion, is that these transporter proteins require a change in shape to work. So here we have the glucose coming in on this side here. We have this change in shape kind of going from a V shape, kind of a teeter-totter kind of movement, to go to an upside down V, moving glucose onto the opposite side of the membrane. And as you can see here, this is still down its concentration gradient, having a higher concentration in the extracellular fluid and a lower concentration of glucose in the cytosol. Now, the rate of carrier-mediated facilitated diffusion is really mostly dependent only on the steepness of the gradient. So here we can see a nice little visual comparing these types of diffusion. So again, this is going to be, we can have this concentration gradient visual here on the left, showing a higher concentration of both, or all three of these molecules, the purple, gray, and gold molecules are all higher concentration on the extracellular fluid side, and a lower concentration on the cytosol side. So they're naturally going to want to move interior into the cytosol. So simple diffusion here, this is going to just be traveling directly through that plasma membrane, not requiring any help and not requiring any energy. Then we have that channel-mediated facilitated diffusion, just kind of like a pore, allowing entrance of very specific molecules through. And then the carrier-mediated facilitated diffusion is going to be those proteins that change in shape to move molecules down their concentration gradient. Next, we have osmosis. So this is a type of diffusion where there is net movement of a solvent through a selectively permeable membrane. So up until now, we've mostly been talking about the movement of solute, so very specific molecules, sodium, potassium, glucose, the movement of those molecules down their concentration gradient. Whereas osmosis is going to be the movement of the solvent, so generally the movement of water and not the solute. So in living systems, like I said, this is mostly water. So osmosis, for our purposes, is going to be the passive movement of water across the membrane and not other molecules. So this is really when molecules of water move from an area of high water concentration to low water concentration. So it's kind of counterintuitive to the other types of diffusion that we've talked about, but we can kind of think of it as the density of water by itself in a small area. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, for example, molecules of water in that small area. Whereas here, in particular, well, similar size, going to be 1, 2, 3, 4, 5, 6, 7 molecules of water for both the same area. This is because there's going to be a lot of other molecules in this area. So it kind of just takes up space, and so not as much water can be in that same area. So we have these solute molecules that take up space. And so here on the left arm here, we have a higher concentration of water as opposed to in the right arm. So water is naturally going to want to move from the left to the right. And so we get this movement, this osmosis, which pushes water into the right arm to create an equilibrium. So now here, if we were to take, for example, a square here and a square here, we would find that the concentration of water molecules would be equal. Okay, let's briefly talk about tonicity. So a solution's tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. Now the tonicity is referring to the concentration of the extracellular fluid. So in this case, the tonicity is the concentration in the water, concentration of solutes. So the first one on the left here is an isotonic solution. The prefix iso means same. So what this is saying is that the concentration of solutes on the outside of the cell here is going to be the same concentration of solutes inside the cell. Now there's always going to be water movement, but the net movement is going to be 0 inside and outside the cell. So what that means is that the amount of water entering the cell is going to be equal to the amount of water exiting the cell. So the red blood cell in this case stays in normal shape. Next, we have a hypotonic solution, and the prefix hypo means lesser than. So what this is meaning is that the solution around the red blood cell is going to have a lower concentration or a lesser concentration of solutes than inside the cell. So we're going to have a much higher concentration of solutes inside the cell. These are just representing molecules and ions. So concentration is much higher inside the cell. And because of that, we're going to have a lot of osmosis occurring. So a lot of water is going to enter the cell here in order to try and equalize the concentration inside the cell and outside the cell. Kind of like we saw on the last slide with osmosis moving in order to reach an equilibrium of concentrations on either side of a membrane. This is what is happening here. So water is going to be rushing into the red blood cell in order to equalize the concentration of solutes inside and outside the cell. And the cell can actually intake only so much water. In this case, when the water content gets too large in the red blood cell, they'll actually burst. They kind of will grow and grow and grow until they burst. And this bursting is termed hemolysis. Hemolysis just means cell bursting. Essentially, they took in so much water and grew and grew and grew until they can no longer handle it, and they burst. Then we have a hypertonic solution. Hyper meaning greater than. So this means that the concentration of solutes outside the cell is going to be very high, much greater than inside the cell. So maybe we only have a very small amount of solutes inside the cell, and outside the cell it's very concentrated. Think of like a very salty water solution. Lots of solutes out there. So because of that, the water is going to want to move out of the cell by osmosis in order to try and equalize the concentrations again. So when water leaves a cell, it's going to shrivel up because too much water leaves, and it is going to undergo crenation. Crenation essentially just means shriveling. So in a hypertonic solution, cells will crenate, and in a hypotonic solution, cells will undergo hemolysis. Okay, now we're going to go over active processes. And active processes require energy in the form of ATP and usually move molecules against their concentration gradients or uphill. So we're first going to look at primary active transport. This is the process in which solutes are moved across cell membranes against electrochemical gradients, and they do this by using energy supplied by ATP. So we have a sodium-potassium ATPase here. This is also known as a sodium-potassium pump. This is a really important example of primary active transport. Before we kind of move into this process, quickly take a look at these gradients. So here we have a sodium gradient, and kind of the darker colors here is just going to indicate a much higher concentration on one side and a higher concentration here. So the sodium is going to be much higher in the extracellular fluid, and potassium is going to be much higher in the cytosol, much higher concentration. So in primary active transport, we have, in this particular case, we have sodium entering the protein here on the cytosol side. So you see it enters in this way. And this is where we have ATP come in. So this is where we have the hydrolysis of ATP. It results in the phosphorylation of the transport protein. Phosphorylation, sorry about the writing there. Phosphorylation, this is going to be adding a phosphate group. So this little green P is representative of a phosphate group here. So this step, the phosphorylation step, causes the protein to change its shape in a manner that essentially pumps the bound solute across the membrane. So we're going to be pumping that sodium out into the extracellular fluid. So this phosphate group causes the protein to change shape, and it changes the opening from the cytosol side here, and it changes the opening onto the extracellular side. Now this pump moves sodium ions outside the cell, and it also moves potassium ions inside the cell. So here we have, when the sodium is expelled, we also have 2 potassium coming in. This causes the phosphate group to be released, and that release of the phosphate group causes this sodium-potassium ATPase to return back to its original shape, and this moves the potassium inside the cell. So as a result of the moving sodium and potassium against their electrochemical gradients, it maintains the gradient. It maintains this electrochemical gradient that is really essential for excitable cells like muscle and nerve cells to function normally, and it's really important also for body cells to maintain their normal fluid volume. Next, we have secondary active transport. Now, secondary active transport is driven by the concentration gradient that we just created in primary active transport. So primary active transport is driven by energy from ATP to move molecules or move substances against their electrochemical gradient. And then secondary active transport uses that electrochemical gradient that was created in order to move other substances. So the sodium and hydrogen gradients created by primary active transport results in kind of this stored energy that can be used to move other substances across the membrane against their own concentration gradients. So here we're going to look at sodium gradient mainly, but the hydrogen gradient is also very useful in secondary active transport. So in secondary active transport in this example, a carrier protein is going to simultaneously bind to sodium and another substance, and then changes its shape so that both substances will cross the membrane at the same time. Couple different types here. We have antiporters and symporters. These are two types of secondary active transport proteins. Antiporters are going to move the substances in opposite directions. So we can see the sodium here is going to move down its concentration gradient from the extracellular fluid into the cytosol. So we can see that movement here. And this particular protein is going to move calcium in the opposite direction against their concentration gradient. So that is an antiporter. Symporters, on the other hand, are going to be proteins that move the substances in the same direction. So again, we have sodium coming down their electrochemical gradient from the extracellular fluid to the cytosol. And we have glucose in this case moving in the same direction, but glucose is going to be moving against its concentration gradient. We're going to talk about another type of active transport which uses vesicles. So first off, to discuss vesicles, vesicles are going to be small spherical sacs within cells. Now, a variety of substances are transported in vesicles from one structure to another within cells. They also will import materials from and release materials into the extracellular fluid. So a couple different terms that we need to learn here. The first one is endocytosis. Endocytosis is when materials are moved into the cells in a vesicle that's formed by the plasma membrane. So move materials into the cells. And then on the other hand, we have another term which is exocytosis. And exocytosis is going to be the opposite of endocytosis. And exocytosis is where materials will move out of the cell by fusion with the plasma membrane, and this is with vesicles that are formed in the cells. So we're going to move materials out of the cell. Now there are three types of endocytosis. Actually, we're going to put this down here. I'm going to write them all the way down here. So the first one is the one actually on this page, which is the receptor-mediated endocytosis. The next type of endocytosis is phagocytosis. And then the last type is bulk phase endocytosis. This one is also called pinocytosis. So the first one we're going to discuss is here. This is receptor-mediated endocytosis. We don't need to know this one in great detail, but we'll just go over some of the major points here. This is where the cells will take up very specific ligands, And this is first by binding to a receptor on the external surface. So this is going to be the external surface of the plasma membrane. And the plasma membrane will actually invaginate to create a vesicle. So we're kind of going to suck in the receptors and whatever particles come with the receptors and create a vesicle within the cell. The vesicle with the materials inside it is going to fuse with a lysosome. Lysosomes are going to be other vesicle types within the cell that contain digestive enzymes. Now these digestive enzymes will kind of digest and break down anything that they come into contact with. So when they fuse with this vesicle containing other materials, it's going to degrade them. And it's going to degrade them into materials that the cell can use otherwise. So it's going to degrade it into amino acids, into fatty acids, and simple sugars. These enzymes will essentially break down these materials that the cell brought in, and it will break it down into usable materials. The next type of endocytosis is phagocytosis, and this is the type of endocytosis where the cell engulfs large solid particles. So this could be really worn out cells, this could be whole bacteria or whole viruses. Phagocytosis is really just engulfing very large particles. So here we have a microbe that is being phagocytized. And there's really only a few body cell types, specifically phagocytes, man, I'm just stumbling over my words, specifically phagocytes that are able to carry out phagocytosis. So this is most likely going to be a phagocyte. That's just going to be the cell type. And this is where we have a particle or a whole bacteria, whole virus, anything like that, this large particle, binds to a plasma membrane receptor on the phagocyte. And once it binds to this receptor, it causes the phagocyte to extend out pseudopods. Now the term pseudopods literally means false feet. So it kind of looks like these little feet that kind of come out from the cell and reach around and grab onto the little microbe that bound to the receptor. Now once a vesicle has been created around this microbe, we call it a phagosome within phagocytes, but it's just a vesicle containing whatever was phagocytized. After we have that vesicle, pretty much the same process happens that we just discussed in the last slide, where we have a lysosome with those digestive enzymes in it. It fuses together, digests everything, and then pretty much the cell uses whatever small materials that were digested by the enzymes. Now, in most cases, any undigested materials in the phagosome will remain almost indefinitely in a vesicle, which is called a residual body. Now, these residual bodies are then either secreted by the cell by exocytosis, they're just kind of spewed out, or sometimes they're going to be stored in the cell in granules. But usually the cell will try to digest everything that's possible so that they can use all these building blocks for whatever the cell needs. Lastly, we have bulk phase endocytosis, also known as pinocytosis. So this is the form of endocytosis in which tiny droplets of extracellular fluid are going to be taken up by the cell. So we can see these little tiny droplets here taken up by the cell, and a vesicle is formed around them from that plasma membrane. There's going to be no receptor proteins involved in this process, and all solutes that are dissolved within this extracellular fluid are going to be brought into the cell. So it's kind of like a cell drinking kind of process because we're taking in those fluid droplets. Again, pretty much the same process is going to happen. The lysosome is going to fuse with that vesicle. Going to digest pretty much as possible and use those building blocks for the cell's needs. Okay, so here we just have exocytosis. We kind of discussed that a little bit before. And here is another term, transcytosis. So this is when the vesicles will undergo endocytosis on one side of the cell. This vesicle will move across the entire cell and then undergo exocytosis on the other side of the cell. Now this most often occurs across endothelial cells, and endothelial cells line blood vessels. And usually transcytosis is a means for materials to move between the blood plasma and the interstitial fluid. Transcytosis also occurs in pregnancy when some of the women's antibodies will cross the placenta into the fetal circulation by transcytosis. Okay, here is a nice little comparison table of some of the passive processes that we talked about. Again, this is just a really good summary table. We've seen these before of these passive processes like diffusion and osmosis, and then the different types of diffusion, simple and facilitated there. Just a little study guide for you. And then here we have a table of the active processes. So looking at primary active transport versus secondary active transport, and then the transport in vesicles that we just went over. So next we're going to talk about the cytoplasm. And the cytoplasm consists of all the cellular components between the plasma membrane and the nucleus. So it includes the cytosol and the organelles, which are the tiny structures that perform different functions in the cell, but it does not include the nucleus. The cytosol, or also known as the intracellular fluid, this is the fluid portion of the cytoplasm that surrounds the organelles, and the cytosol makes up about 55% of the total cell volume. The cytosol does vary in composition and consistency from one part of the cell to another. Generally, it's about 75 to 90% water, plus various dissolved and suspended components. So these would include things like ions, glucose, amino acids, fatty acids, proteins, lipids, ATP, even waste products are going to be dissolved within the cytosol. The organelles, on the other hand, these are going to be these tiny little specialized structures. They each have their own very specific shape and have very specific functions. We will talk about most of the organelles each individually. So here we just have a nice little summary table going over the description and functions of different parts of the cell, including the plasma membrane, the cytoplasm, which includes the cytosol, and organelles. Also going to have a lot of other organelles here that we will discuss a little bit later in this chapter, including the endoplasmic reticulum, or the ER, Golgi complex, and the mitochondria. And this table also shows the nucleus as well. So first we're going to discuss the cytoskeleton before we go into any organelles. So the cytoskeleton is a framework of protein filaments that extends throughout the cytosol. Now there are three types of filaments that contribute to the cytoskeleton structure, as well as the structure of the organelles. We're going to go over them in order of their diameter, starting with the smallest diameter is a microfilament. Then next largest is an intermediate filament, And then the largest diameter is a microtubule. So first we have the microfilaments. These are the thinnest elements. They are composed of actin and myosin. And these are going to be most prevalent at the edges of the cell. So here we can see just in this little picture here, these microfilaments are going to be mostly prevalent just along the edges there. Now they do serve two general functions. The first one is to help generate movement. And they also provide mechanical support or physical support. Now, with respect to movement, they help in things like muscle contraction, cell division, and also cell locomotion, or the whole cell moving locations. Now, like I said, they also do provide mechanical support. Now, this would be support for things like cell extensions like microvilli. So you can see here, these actually go into the microvilli too. These are going to be, these microvilli are non-motile, so they don't move. They are very microscopic, finger-like projections of the plasma membrane, and the microvilli really help to increase the surface area of the cell. So the microfilaments within the microvilli help to provide that mechanical support, help them to keep their shape. Next, we have the intermediate filaments. These are going to be a little bit thicker, and they are exceptionally strong. Intermediate filaments are found in areas of the cell that are subject to mechanical stress. So they really help to stabilize the position of organelles, such as the nucleus in the middle there. And they also help to attach cells to one another. So I'll put here, we're going to say places. So they're found in places where there's mechanical stress, mostly for stabilization. Like helping the nucleus be stabilized here. Next, we have microtubules. These are going to be very long, unbranched, hollow tubes. And these are composed mainly of the protein tubulin. Now, the assembly of microtubules begins in an organelle called a centrosome, and they grow outward toward the periphery of the cell, or toward the plasma membrane. Now, the microtubules help to determine the shape and also the function in the movement of organelles like secretory vesicles, chromosomes during cell division, and specialized cell projections like cilia and flagella. So we're going to say they help the shape, and well, shape and movement. We'll put shape and movement of organelles like secretory vesicles, chromosomes. This is just during cell division. And then specialized cell projections like cilia and flagella. Okay, now we're going to start talking about specific organelles. So as we kind of briefly mentioned earlier, organelles are going to be specialized structures within the cell that have very characteristic shapes and perform very specific functions. Now, each type of organelle has its own sets of enzymes and carries out specific functions and serves as a functional compartment for a very specific biochemical process. Now, the number and types of organelles varies in different cells, and this really depends on that specific cell's function. But really, organelles all cooperate and work together to maintain homeostasis, even though they have very different functions and very different specific functions. So first off, we are going to talk about the centrosome, and we definitely briefly talked about this one just earlier when we talked about microtubules. So centrosomes are generally located near the nucleus, and it consists of a pair of centrioles and also pericentriolar material. The two centrioles, these are going to be very cylindrical structures and not too important on the specific details, but just for interest's sake, they are each composed of these 9 clusters of 3 microtubules arranged in this circular pattern that you can see here. You don't have to remember this for an exam or anything, but these centrioles are going to be the organizing centers for the growth of the mitotic spindle. And we'll learn it a little bit later, but you might have learned this also in high school biology, that the mitotic spindle plays a really critical role in cell division. And this is the actual structure that pulls the chromosomes to each pole during cell division. These are also going to be the organizing centers for microtubule formation in non-dividing cells as well. Now, microtubules are the dominant components of cilia and flagella, and these are motile projections on the cell surface. So cilia, these are going to be, we can see them right here, these little green little projections on this picture here. This is labeled cilia. These are very numerous, but they're going to be quite short hair-like projections that extend from the cell surface. They each contain about a core of maybe 20 microtubules that's surrounded by a plasma membrane. So you can kind of see this here with the microtubules all here surrounded by plasma membrane, kind of the same structure that we see from a cilia or a flagella here. The basal body, this is the part that anchors all the microtubules together just below the plasma membrane surface. Flagella, like I said, they're going to be really very similar in structure to cilia, but typically flagella are very much longer, and usually flagella work to move an entire cell, like we see here with a sperm cell. So this is a flagella moving the entire cell, whereas cilia are more just little hair-like projections that usually just move things along the surface of the cell, like move mucus along the surface of the cell, and don't move entire cells locations. So here we kind of have the movement of both cilia and flagella. The top one here is the cilia. So a cilium displays kind of an oar-like pattern of beating. So it's relatively stiff during the power stroke, or going from left to right in this picture, but more flexible during the recovery stroke. So you can kind of see on the recovery stroke, it's very bent and going back, and on the power stroke, it stays very straight, kind of this lighter color back here. Now, many cells of the respiratory tract have hundreds of cilia, and they're mainly used to help sweep foreign particles that are trapped in mucus away from the lungs. In cystic fibrosis, it's kind of interesting because cystic fibrosis produces extremely thick mucus secretions, and this thick mucus interferes with the ciliary action and also interferes with the normal functions of the respiratory tracts. Down here we have the flagellar movement. Flagellum generate forward motion, and this is by rapidly wiggling, kind of this wave-like pattern that we see here. The only example of a flagellum in the human body is a sperm cell's tail, and this is going to propel the sperm toward the oocyte in the uterine tube. Next, we have ribosomes. These are going to be the sites of protein synthesis. Now, their name reflects their high content of one type of ribonucleic acid, which is called ribosomal RNA, or also called rRNA. But each one of these ribosomes also includes more than 50 proteins. So it's made up of rRNA and also a lot of proteins. Now, structurally, the ribosome is made up of two subunits. We have a large subunit and a small subunit. The small subunit is approximately half the size of the larger one. And these two subunits are made separately in the nucleus, and then they exit the nucleus separately, and then they come together in the cytoplasm. So these are made in the nucleus separate. Now, ribosomes can actually be found in a couple of different places. They can either be attached to other organelles, such as the outer surface of the nuclear membrane, the membrane on the nucleus, or they can also be attached to an extensively folded membrane called the endoplasmic reticulum. So I'll put here they can be attached to the nucleus, or they're also found on the endoplasmic reticulum. We'll just abbreviate that ER, but that's endoplasmic reticulum. Or they can also be free. This is just meaning that they are unattached to other structures in the cell. They're just kind of free floating in the cytosol. So you can either find them attached or free. Free ribosomes are used to synthesize proteins that are used in the cytosol. So the proteins that are made by ribosomes, just that are free floating, they are used in the cytosol themselves. There are also ribosomes located within the mitochondria. But these are just used to synthesize mitochondrial proteins, so proteins that are used only in the mitochondria. So next, we're going to talk about the endoplasmic reticulum. Now, this is a network of membranes in the form of flattened sacs or tubules, and it extends from the nuclear envelope. Remember, that's the membrane around the nucleus, and it projects throughout the cytoplasm. The endoplasmic reticulum, or also known as the ER, it's a very extensive network, and it makes up more than half of the membranous surfaces within the cytoplasm of most cells. Now, there are two types of ER within cells. There's going to be the rough ER, or the rough endoplasmic reticulum, and the smooth ER. So first off, the rough ER is going to be continuous with the nuclear envelope. And it's usually folded into a series of folded sacs. So we can kind of see these little fold structures here. And that's what the rough ER is going to look like there. The outer surface of the rough ER is also studded with ribosomes. So we can see those here, kind of blue in this picture. And these ribosomes are going to be the site for protein synthesis. Proteins that are synthesized by the ribosomes attached to the rough ER, they enter the spaces within the rough ER and they are processed and sorted. Okay, now the proteins that are made by ribosomes in the rough ER are going to be secretory proteins or membrane proteins, proteins that are put out onto the surface of the cell. These are going to be kind of stuck on the membrane of the cell. And then there's going to be a lot of other organellar proteins. Proteins that are used by other organelles. Okay, then we have the smooth ER, and this one extends from the rough ER and it really just forms a network of these membranous tubules. So you can kind of see here the rough ER is very flat and kind of folded, whereas the smooth ER kind of forms more of these little tubes. So it does look quite different from one another. Now, unlike the rough ER, the smooth ER does not have any ribosomes on the outside surface, but the smooth ER does contain unique enzymes that make it a lot more functionally diverse than the rough ER. Now, because there's no ribosomes on it, the smooth ER does not synthesize proteins, but it does synthesize things like fatty acids, we'll say acids. I'll rewrite that here. Fatty acids, also steroids. And these are going to be steroids like estrogens and testosterone. Next is the Golgi complex. This is also called the Golgi apparatus. We'll just put AKA, also known as. Now, this has a special function as well, because most of the proteins that are synthesized by the ribosomes attached to the rough ER are going to be transported to other regions of the cell, but the first step in this transportation is going to be the Golgi complex. So these are going to be proteins made by rough ER ribosomes are going to come to the Golgi apparatus. So this is going to be the first step after those proteins are made in the rough ER. They're going to come to the Golgi complex. Now the Golgi complex has a couple of functions. So what it mostly does is it's going to package proteins into nice little vesicles. We can see these here. And it's also going to modify proteins. So these are going to be kind of the main functions of the Golgi complex. Now the Golgi complex is made up of 3 to 20 of these cisternae. Now cisternae are going to be very small, flattened sacs that have these bulging ends, you can see here. Now, cisternae at the opposite ends of the Golgi complex will differ from each other in size and shape, and also in the enzymatic activity. So this would be the cis phase versus the trans phase. So this is where they enter in the cis phase on this side, and they exit here on the trans phase. So the cis phase versus the trans phase, like I said, they have a difference in size and shape and also different enzymes between them or within them. So the cis face is going to be this convex surface, and this is where it's going to face the rough ER, and it accepts the proteins that are synthesized by the ribosomes attached to the rough ER. This is where those vesicles that have proteins made by the rough ER ribosomes come in. And they will travel through all these middle cisternae. So they will move this way, traveling through all these cisternae. And as they travel through these folds in the Golgi complex, there's different enzymes and different structures within the Golgi complex that will modify these proteins, package them differently until they reach the trans phase. And then they are secreted out. And now that they are modified and packaged properly to go where they need to go. So here we can see the Golgi complex in a little bit more detail. We can see here the rough ER on the left side with the ribosomes attached to that rough ER. This little orange squiggle here we can see is a synthesized protein that's been synthesized by those rough ER ribosomes, and it gets put into a nice little transport vesicle. That transport vesicle will travel through the cytoplasm into the entry phase cisterna. So we can see number 3 here, that transport vesicle is endocytosed into the Golgi complex there. Now this little protein will travel through the medial cisternae. This is where carbohydrates are added, glycoproteins, lipids are going to be added to these proteins as they travel through the Golgi complex. And then the exit, or the trans phase, this is going to modify these molecules a little bit further and then sorts and packages them to transport them to their very specific destinations. So here we can see one is going to lysosomes. This one labeled number 8 is going to be sent to the plasma membrane. And then this one's going to be secreted over here. So these proteins are secreted, exocytosed from the cell because they are secretory proteins, proteins that are secreted out. Things like hormones would be secreted. Here, these proteins are built into the plasma membrane, kind of merged into the membrane. And this vesicle here, this transport vesicle, is going to stay within the cell. Next, we have lysosomes. These are going to be membrane-enclosed vesicles that form from the Golgi complex. Remember, we just saw on the right side of that last diagram with the Golgi complex, those vesicles that stay within the membrane, these are going to be lysosomes. And they contain as many as 60 kinds of these very powerful digestive enzymes. And these enzymes break down really a wide variety of molecules once these lysosomes fuse with vesicles that are formed during endocytosis. Now, lysosomes mainly work to break down proteins that come to them in the form of vesicles. So they work to break down molecules in other vesicles when they fuse together with those other vesicles. Now, Lysozymal enzymes work best at acidic pHs. Now, within these membranes here, there's going to be active transport pumps. So these are going to be active transport pumps that are going to be constantly importing hydrogen ions. And this allows the interior of lysozymes to have a pH of approximately 5. So this is going to be very, very acidic. It's about 100 times more acidic than the pH of the cytosol, which is 7. So it means kind of like in this picture up here, we have a pH of 7, and within the lysosomes, we have a pH of 5. So very, very acidic. Now, pretty much the lysozymes kind of act like the trash bins of the cell. They are going to take in damaged or unneeded organelles, debris, or really anything else, any garbage that the cell has, and it's going to digest it completely. And then the lysozymes are going to release the usable molecules back into the cytosol. So it's going to things like glucose, amino acids, and fatty acids. These are all going to be broken down molecules that can be used by the cells for other things. Next, we have proteasomes. Now, as you can probably guess from the prefix proteal, these are going to break down proteins. But unlike lysosomes, which also break down proteins, proteasomes degrade proteins that are found free floating in the cytosol. Now, for example, proteins that are part of metabolic pathways, they need to be degraded after they have accomplished their function. Now, these protein destruction pathways, like what a proteasome does, plays a negative feedback into halting the pathway once the appropriate response has been achieved. So essentially, the proteasome is going to find proteins that have completed their function and break them down so that those proteins don't continually to try and complete their function over and over and over again when they don't need to. Once they're finished it once, they can be broken down. They are finished with their role. Proteasomes are really small and they chop up proteins into amino acids, which can then be used by the cell to be recycled into new proteins. Essentially, lysosomes and proteasomes just work as recyclers, essentially. I know I say garbage bins for lysosomes, but really it just breaks everything down into its smallest unit to be reused by the cell and recycled into other new fatty acids, new proteins, new carbohydrates. Now, the next organelle we're going to discuss is the mitochondria. This is known as the powerhouse of the cell, and this is because most of the energy in the form of ATP is going to be created in the mitochondria, and this is usually through aerobic production, aerobic respiration, which we will discuss later. Aerobic respiration is going to mean that it requires oxygen. Now, a cell may have as many as a few hundred or even several thousand mitochondria within it. Now, a little bit just on the anatomy of the mitochondria. It has an outer membrane and an inner membrane, so two membranes, and there's going to be this small fluid-filled space between those two membranes. There's going to be these really elaborate folds that we call cristae that provide a really enormous surface area for chemical reactions that are part of the aerobic phase of cellular respiration. And remember, these are going to be the reactions that produce most of a cell's ATP. Remember the ATP, this is going to be the energy for a cell. Now, the mitochondria also serves a really important role in apoptosis. This is also just another term for programmed cell death. Now, apoptosis can occur in response to many things. Could be destructive free radicals, damage to DNA within the cell, could be a lack of oxygen and nutrients, and all these reasons would result in certain chemicals being released from the mitochondria, and this creates a pore in the outer mitochondrial membrane. Now, one of these chemicals is known as cytochrome C. And generally, this is in the mitochondria pretty happily. It works in creating ATP. But if cytochrome C is released into the cytosol, it initiates this cascade of activating protein digesting enzymes that bring about apoptosis. So we're going to learn a lot about this a little bit later, but essentially we know that the mitochondria can essentially kill the cell if certain conditions are met. If certain things happen, like there's a lack of oxygen and nutrients, the mitochondria just kind of flips a switch and releases cytochrome C into the cytosol, and this brings about cell death or apoptosis. Now, a cool thing about the mitochondria is that it has its own set of DNA. This is called mitochondrial DNA. And this is DNA that's completely separate from the nucleus. Now, genes from the nucleus, these are going to be inherited from both your father and your mother, but mitochondrial DNA is only inherited from your mother. Now moving on to the nucleus. This is a spherical or sometimes oval shaped organelle. It's usually the most prominent feature of a cell. Now most cells will have just one single nucleus, though there are a couple exceptions. For example, red blood cells, they have no nucleus. And skeletal muscle cells and some other cell types, they will have multiple nuclei in each cell. Now there is a double membrane that encloses the nucleus. So you can kind of see the nuclear envelope here, a little bit of a zoom in. We have kind of this inner membrane here and an outer membrane here, and there are pores throughout that double membrane. Now these nuclear pores help to control the movement of substances between the nucleus and the cytoplasm. Small molecules and ions can move through the pore through passive diffusion. Remember, they'll have to move down their concentration gradient, like we discussed before. But most larger molecules, like RNAs and proteins, they can't pass through directly through the pores. They actually have to pass through using active transport. So it's only usually small little ions and molecules that can directly pass through these nuclear pores. Now within the nucleus, we have the hereditary units called genes, which can be condensed into chromosomes. So here we have the DNA double helix, and this is going to have the instructions for all the genes that make up your whole body. So for example, we could have maybe from here to here on the DNA, this could hold the instructions for eye color, for example. So this DNA double helix, it's going to have linked nucleotides that are going to twist around histones, which are going to be proteins. And those histones are going to twist together and further twist and compact into nucleosomes, and you can kind of see they even compact and twist even more here and continue twisting until they kind of sort of resemble this bow tie. This bow tie structure is called a chromosome. Now, as you can see, because it's so compacted, there's going to be really a ton of DNA within these chromosomes. Now, DNA encodes for everything in the cells. Pretty much, DNA is the blueprint for all the proteins that a cell needs to survive. Now, in a process called gene expression, the gene's DNA is going to be used as the template for the synthesis of a very specific protein. So first, kind of step one, the DNA is going to be transcribed. Now, transcription essentially is a process where DNA is going to be rewritten into RNA. Specifically, DNA is going to be converted or rewritten into a specific type of RNA called mRNA, otherwise known as messenger RNA. Now, that copied mRNA molecule is going to be transported into the cytosol, or into the cytoplasm here, and it's going to be transported to a ribosome. Now, the RNA is going to attach to the ribosome, and this is where we have step 2, where the protein is going to be translated from that copy of DNA. So we'll go over transcription and translation in a little bit more detail here. So the first step being transcription. This is where DNA is going to be unwound. So we're going to kind of unwind that double helix, and the DNA is going to be copied into a different but more readable form. So this form is called mRNA, like I said. So it's going to be kind of converted or rewritten into mRNA. Remember, this is messenger RNA. And nucleotides, which make up DNA and RNA, are used to transcribe the DNA code into mRNA. Now, there's a lot of modification that occurs on this mRNA molecule, as the entire transcribed portion doesn't actually code all for a protein. So when a long portion, we won't actually go into too much detail about this, but say there's a long portion of DNA that is transcribed into mRNA. So we'll say this is going to be the DNA portion, and it's going to be converted into mRNA. Now, there's going to be sections within here that are called introns. So maybe from here to here, this is an intron, and choose different color here. Maybe from here to here, this is an exon. Now, exons, these are going to be the portions that actually code for proteins. So this is going to be the actual protein code. And introns, these are kind of just filler code. Doesn't actually code for any particular protein, just kind of filler RNA. So it doesn't actually code for proteins that we know of. It could do something else or have another function, but we kind of don't think that it codes for any type of proteins. Now, there are very specific enzymes that will cut the introns out of the mRNA so that we're just left with that functional protein coding code that's all left over. So that we cut out the introns, we're just left with exons. Now, all of this processing all occurs in the nucleus before the mRNA is sent out of the cytosol. Now, this is kind of just a brief overview. You don't need to remember all these specifics, but just know that the exons in mRNA code for proteins, and mRNA is just going to be that more readable version of the DNA code. Now, the second step in protein synthesis is going to be translation. Now, translation occurs in the cytosol. This is after the mRNA has been transcribed from DNA, and we've also modified that mRNA so that we're removing all those introns. We've sent it out of the nucleus into the cytosol, and then this is where we get to translation, the actual protein synthesis part of this entire process. So this is where the mRNA comes to ribosomes, and the ribosome will kind of read it, and it will tell the ribosome which protein to make. So here we have this long mRNA segment. We can see right here is labeled, and it first is going to attach to the small subunit of a ribosome. And then the large subunit is going to join together, and essentially the large and small subunits will read the mRNA, and it'll tell the ribosome rather, which amino acids to bring in. The ribosomes will use these other type of RNA, which is called tRNA. This is the transport RNA. And tRNA is going to be the actual structure that brings amino acids to the ribosome for building proteins according to the mRNA strand. So say that the mRNA has a code like AUG. AUG, for example, would code for the amino acid methionine. And so pretty much the mRNA is going to tell the ribosomes to bring methionine because we're coding for methionine. This is what the instructions say. Instructions say methionine is going to be the first amino acid in the sequence. And so the ribosomes will talk to tRNA and tell tRNA, hey, bring methionine. That's going to be the first instruction that this mRNA is telling me to build. And so it will continue in that process until we build more and more amino acids, kind of making this chain. That's kind of the gist of it. We don't need to know entire process of transcription. The main thing is that we need to know is that mRNA brings the instructions to the ribosomes, and the ribosomes will read the instructions and tell tRNA which amino acids to bring in order to build those amino acids into a chain, creating the protein that mRNA codes for. So we've kind of gone over all the organelles in the cell, or at least all the organelles in the cell that you need to know for this class. So now we're going to briefly go over cell division. So mitosis occurs when somatic or body cells need to duplicate. Somatic cells is just another word for body cells. So we're going to talk about mitosis. So human cells contain 23 pairs of chromosomes. And so this is a total of 46 chromosomes completely. And some cells will divide more than others. When a cell reproduces, it must duplicate all of its chromosomes to pass on its genes to the next generation of cells. So cell division is really just that process in which cells reproduce. So we can kind of see the cycle here on this picture on the right. Now, most of the cell cycle consists of what is called interphase. This is in the light purple color here. And interphase is really just the preparation for cell division, but it's not dividing yet. So here, this is really just division preparation. So interphase is kind of more divided into 3 different phases. This would be the G1 phase, the S phase, and the G2 phase. Pretty much G1 phase is where the organelles and cytosolic components are duplicated. G2 phase, this is where pretty much like the enzymes and other proteins are going to be synthesized. And then our S phase is going to be our major phase where DNA is all replicated. Mitosis down here, or the mitotic phase, this is when the cell is actually dividing. So this is going to be the dividing phase, and it is broken up into, we call it PMAT, P-M-A-T. This is also known as prophase, metaphase, anaphase, and telophase. Now, cells will always start into mitosis, kind of right here. This is where they're always going to start as a diploid cell. This term diploid means that the cell contains 2 complete sets of paired chromosomes, or duplicated chromosomes. I'll just put dup-chrome for duplicated chromosomes. So this is when all of their DNA has been replicated, and they're coming into the beginning of metaphase with completely like 2 sets of everything. Then they're going to go through this mitosis where we have 1 cell is going to divide into 2 cells. And so we're going to end up with 2 haploid cells. Haploid is a term where it means that these cells have a single set of unpaired chromosomes. We also term diploid 2N, where we have 2 sets of genes, and haploid just being a single N, which means it is a single set of genes. So here is just showing we have interphase being broken up into G1, S, and G2. Interphase, we pretty much have all of this chromatin. This is just the non-condensed version. This is when DNA is all just kind of spaghetti looking. This is kind of non-condensed DNA. So this is when DNA hasn't been completely coiled up and turned into those chromosomes. It's just kind of uncoiled. It kind of fills the entire space within the nucleus. Now we get into mitosis. Now, mitosis is really an exact partitioning of genetic information into 2 identical cells. So there are really 4 stages of mitosis, remember that PMAT, but this is a continuous process where each phase kind of seamlessly merges into the next. So remember, during that interphase, we had this very spaghetti-like, non-condensed DNA material, but during prophase, this first phase of mitosis, the chromatin fibers are going to really condense and shorten. And they're going to shorten into chromosomes that are going to be very visible under light microscopes. So chromatin, you can't really see it very well under a microscope because it's all just very spread out and isn't really condensed enough to see. But once it's condensed into chromosomes during prophase, we can actually see them under a microscope. Now, because that longitudinal DNA replication took place during the S phase of interphase, each prophase chromosome consists of a pair of identical strands that are called chromatids. So each chromosome consists of 2 chromatids that are joined at a centromere. This is going to be this constricted region that holds the chromatid pairs together. So here, I'll just draw a little picture here for you. We have one chromatid here. I'll draw in a different color, but these chromatids are going to be identical to each other, kind of. And in the center here, this is where we have the centromere, this constricted region holding those chromatids together. And just outside this centromere, kind of like right here, we have this protein complex that is known as a kinetochore. You can see this is labeled here. Now, a little bit later in prophase, kind of here we have early prophase and late prophase. This is when tubulins in the centrosomes are going to form the mitotic spindle. So here we have those centrosomes in green. Kind of talked about them a little bit before. We have these mitotic spindles growing off of those centrosomes. Now those mitotic spindles are actually going to attach to the kinetochores of the chromosomes. So we can see this here. We have one mitotic spindle coming here and attaching to the kinetochore there, another one here attaching to this kinetochore, so on and so forth. Now, kind of also what happens in late prophase is that we have the nucleus disappear, or the nuclear envelope disappear as well, and also the nucleolus disappears. So we can kind of see in this left picture, we have that nuclear envelope around here. Still starting to break down in this picture, but here in late prophase on the right side, we have no nuclear envelope. Now that next phase of mitosis being metaphase, this is where the microtubules of the mitotic spindle will align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. So we have mitotic spindle going from here to here, and then here at the exact center, we call that the metaphase plate. So the chromosomes will be lined up at the metaphase plate during metaphase. Now during anaphase, the centromeres are going to split, separating the 2 members of each chromatid pair. So see you have one chromatid here and one chromatid here. They're going to be pulled to opposite poles of the cell. Now once they are separated, the chromatids are actually now each individually called chromosomes. So you can see this is labeled here. So this one's a little backwards. It has early anaphase on the right, late anaphase on the left here. But those chromatids are going to get separated, and once they are their own unique structure, they're called chromosomes. And as they're pulled apart from each other, the cell starts to kind of divide itself, kind of separate itself into the beginning of 2 cells. And this is by a cleavage furrow. So it's kind of starting to constrict just a little bit in the middle here, the beginning stages of separating into 2 distinct cells. Now the final stage of mitosis, which is telophase, this phase begins after chromosomal movement has stopped. The chromosomes are now in their respective poles, and they're going to uncoil and revert back to that thread-like chromatin form. So they're going to regain their chromatin appearance. We're also going to see a new nuclear envelope or a new nuclear membrane form on either side. So we can see this here on both sides. And these mitotic spindles will start to break up. We'll get also to get a little bit deeper of a cleavage furrow here. But even at the end of telophase, the complete separation of these two cells is not finished yet. This is where we get to cytokinesis. So cytokinesis, this is a term given to the division of the cell's cytoplasm and organelles into two identical cells. So this entire process of cytokinesis usually begins in late anaphase with that beginning formation of the cleavage furrow, and cytokinesis is completed after telophase. So here we can see anaphase. This is when we're starting to have cytokinesis begin with that beginning of the cleavage furrow. In telophase, a little bit more of a cleavage furrow. But then after telophase, cytokinesis is finished when these cells are completely separate. So we can kind of have this process be summarized like this. So we have the G1 phase. G1 goes into S phase. S phase goes into the G2 phase. G2 phase goes into mitosis. And mitosis go into cytokinesis. But again, some of cytokinesis occurs during mitosis as well. Here's a nice summarizing table of all the events of a somatic cell cycle. So again, somatic cell just means all of your body cells. Just includes all the phases we discussed and the activities of those phases. Now there are three possible destinies for every cell. They can remain alive and just function without dividing. They can grow and divide, or they can die. So these are going to be our essentially three fates that can happen to any cell. Okay, we are going to very briefly go over meiosis, which is a little bit different from mitosis, but just a few differences between them. Now, reproductive cells or gametes will go through meiosis. This is also known as reproductive cell division. And reproductive cell division occurs in the gonads. So this is in the ovaries and the testes. Reproductive cell division produces gametes. So these are sperm and ovum. And each of these contain a single set of 23 chromosomes. Fertilization of the two gametes, one sperm and one ovum, will restore the diploid number of chromosomes. Now, unlike mitosis, which is complete after a single round, meiosis, on the other hand, has two successive stages, meiosis 1 and meiosis 2. So meiosis 1 begins once chromosomal replication is complete, and it consists of 4 phases. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Now there's this process called crossing over that occurs during prophase 1. And this allows there to be an exchange of genes between chromatids of homologous chromosomes. So you can kind of see right here that we kind of get this crossing over, and then we get two completely different chromosomes here. So the resulting cells from this crossing over allows the cells to be completely genetically unlike each other and completely genetically unlike the starting cell that produced them. Homologous chromosomes, or the same paired chromosomes, will pair up along the midline in prophase 1. And then they go through this crossing over here. And then they'll go into anaphase. They go through metaphase first, but this is just when they kind of pair up along the metaphase plate. And then in anaphase, the members of each homologous pair of chromosomes will separate, and they're going to be pulled to each pole. So you can kind of see here we have these kind of mixed up chromosomes now with a little bit of blue and a little bit of red on them. So they're very different from the original chromosomes up here in prophase. So this kind of separation that we see here in anaphase 1 is actually different from mitosis. Remember, in mitosis, the centromeres will split in anaphase and the sister chromatids separate. So here we have full chromosomes, full bow tie chromosomes like this, that will actually be pulled to each individual pole. So this one goes that way and that one gets pulled that way. Instead, in mitosis, we would have this chromosome, or chromatid, I guess in this case, get pulled that way and this way. This was in mitosis. So it's a little bit different between meiosis 1 and mitosis, even though they both still occur in anaphase, but anaphase is a little bit different between the two. So the net effect of meiosis 1, which is just going to be this one, this is going to be resulting in every cell containing a haploid number of chromosomes, because it contains only one member of each pair of the homologous chromosomes that are present in the starting cell. So we're going to end here with 2 haploid cells. Meiosis 2 is essentially the same steps, and it's essentially the same, but meiosis 1 starts with a diploid cell and ends with 2 haploid cells, and meiosis 2 starts with 2 haploid cells in prophase 2, which will each divide and result in 4 haploid gametes that are genetically different from the original cell. So here we have four haploid gametes resulting at the end of meiosis 2. Also, one other thing to note is that that crossing over that we saw earlier only occurs in prophase of meiosis 1. So it only happens in prophase 1. We don't see that same crossing over occur in prophase 2. Now, as we age, there are a few things that happen to our cells. Just very briefly, they start to deteriorate in their functioning. So they can't function as well as younger cells. The actual number of body cells decreases. It's because the body cells will die as they get older and they're not replaced by newer cells. And also the integrity of the extracellular components of tissues deteriorates. We also get free radicals more as we age. So a free radical is an uncharged molecule. Typically, they are very reactive as they have unpaired valence electrons. So in the body, these can actually be quite destructive. And that's going to be the end of chapter 3.